Okay, well, I'd like to welcome everybody to the January 20th meeting of the IMAG MSM Working Group on Multiscale Modeling in Viral Pandemics. I'm pleased that we have two great speakers lined up for today. I'd like to remind you that the meeting is being live streamed on YouTube and is being recorded, so please keep that in mind. And these recordings are available after the fact for people who miss the meeting in person. People have seen this slide many, many times, but there is a change to the slide for people who have seen it so many times. Reinhardt and I continue as co-leads. Jim Sluka is going to continue in web administration, and there is going to be a gradual handover between Bruce Shapiro, who's done such incredible work over the past year and a half on this working group, with Lorenzo Veschini from London, who's been one of our loyal participants over the years. And so you'll be hearing, I think, more from Lorenzo about the administration of the working group going forward. I want to thank Bruce again and also Lorenzo for stepping in. We have our usual list of communication channels. They only work if you use them. Please do help us out. Again, I want to call attention to our great archive of talks over the past year and a half. It's a tremendous resource. Please help us with those to make sure that people are aware of them. We have a Twitter handle. People can use that. Help us retweet the announcements of the workshops and make sure that our speakers get the audiences that they deserve for their work. Are there any announcements for today? We have a steering committee meeting tomorrow morning. Are there any other announcements to make at this point? Reinhardt, do you have anything you want to?
No, I don't. Thank you.
No? Anyone else? No? Well, in that case, I want to come to our schedule for upcoming meetings. Next week, we have two talks scheduled. February, we are a little short. So I urge you to volunteer yourself, your students, your postdocs, or send us the names of the people you would most like to hear from, and we will try to get them on the schedule. In particular, it may be hard to bring in outside participants for February 3rd, so we would love to have somebody step up and volunteer to speak for February 3rd. If you think you volunteered and somehow didn't get a response, please contact us again to make sure that we can get you on the schedule. Again, it's a great opportunity if you have senior graduate students or postdocs who want to present their work and get some exposure. We have a lot of very senior people, but we also have a lot of more junior people speak. Please don't be shy about speaking or speaking again if you have already spoken at some time in the past in the working group. We'd also be happy to hear from subgroups and subcommittees. If you want to report on your progress, please let us know and we'll get you on the books. So please do help us out in terms of schedule. Remember, please to mute your microphones. Both of our speakers have very generously agreed to stay for discussion afterwards. So the format will be, we'll have the two brief mini presentations, and after those are over, we will have questions and discussion with both of our speakers. We appreciate very much our speakers being willing to stay for the discussion session. People know that I don't do elaborate introductions. We're very pleased to start with Catherine Beauchemin from Ryerson in Toronto, who's going to be talking about isolating and quantifying the efficacy of virus replication steps. And I will turn it over to you.
Hi, all right. Let me go ahead and share my screen. Can you hear me well? Okay, perfect. Share, this should work. You should now be able to see it. Is it blurry or is it a good resolution? I'm gonna take it to mean it's good resolution?
There is a black box, which is probably the Zoom controls.
Yeah, let me make this a little bit better. I can fix that here. It should be, wait, no, not quite. Okay, just
that looks fine to me.
it's gonna keep messing up. I'm just gonna rewire this a bit. This should then be perfect now. Is the resolution okay? Everything is crisp? Okay, so
That
I'm
looks good.
Perfect. Okay, so
Are
I'm
you
gonna
going
go
to
ahead.
tell us why you have katakana on your title?
Yes, I am currently based in Japan. Oh, I have not turned on my video, so in case you want to see my face, hi. I am currently based in Japan, and it's 5 a.m. here, so I'm gonna.
Oh, I'm sorry, I didn't realize that you. Okay.
No worries. So basically, I'm going to try and not speak too loud for my neighbors. And the other reason for the X on my university in Toronto, you can maybe read the news if you want to know more about our renaming process at Ryerson. So yes, indeed, I'm going to talk about how you can isolate and quantify individual virus replication steps by fine-tuning the experiments that are done. And this is research that's funded by the following organizations. And it's a bit of a generic talk because the time is fairly limited, but I'm happy to answer questions that are more specific afterward. The first thing I want to do, though, is to advertise our Physics of Complex Systems program at Ryerson. And so these are my other three colleagues here, Aidan, Sean, and Eric, along with myself. We're the 4 members of this new research field at Ryerson University. It's a graduate level research field. So if you're looking for a graduate program, maybe check us out. So with the advertisement out of the way, let me explain how we kind of break down a viral replication in terms of parameters first. And there will be no math in this talk. I've been asked to keep it for a general audience. You would start with an uninfected cell, and the cell could become infected as virus enters the cell, and it would enter what is called the eclipse phase, where the cell is infected but not yet producing virus. After some amount of time, tau e, the cell would begin to produce virus. The virus would then be released into the medium. And after some amount of time of the cell producing that virus, and it could produce it as a burst or as a continuous production, depending on the type of virus, because that depends from virus to virus, the cell would eventually then die or at least cease virus production. The distinction between death and ceasing virus production is not that relevant for us. It's important whether the cell is a producer or can be a source or a sink. Once it can be neither, it's quite unimportant. And because we tend to represent in vitro system, the idea that the cell could be replaced by a new, fresh, uninfected cell is not relevant for our simulation. So we tend to simulate and capture an in vitro experiment. And so the infectious cell then produces virus. That virus either goes on to infect other cell, or it can be lost through loss of infectivity. So inside a person, that virus could be bound by antibodies, or could be just lose infectivity or physically removed, for example, through your cilia, that could be beating and removing the mucus. But in vitro, basically, the virus has nowhere to go. So the only way we lose virus is it's still there, but it loses the ability to infect cells. So this is the rate at which the virus loses infectivity. So this is basically the main parameters of our model. And you might ask yourself then, like, how can we find the value of each of these parameters? So we've done that firstly by simulating this model and simulating various experiments with it to see which experiment could give us the information we need. And the first one would be, of course, the viral decay assay. So this is where you have no cells, just virus in a well. And what you do is you count the virus only as a function of time. So you take a sample at 24 hour, 48, 72, and then you'd be counting how much, for example, infectious virus, so maybe in units of TCID 50 or PFU or FFU, and you'd be counting how much virus you count as a function of time. And because it's losing infectivity, you would count fewer and fewer virions as a function of time, infectious virions. And then you could also do the same thing, but instead of counting by black assay or by TCID50 assay, you could be counting by QRT-PCR. And so in that case, you could count how many virus balls are left as a function of time. And 84 hours is not very long, but we still see some decay. And so you can estimate even the rate at which the virus will lose enough integrity that it's no longer counted in a QRT-PCR assay. So with just this experiment already, we can characterize parameter C. So that was this parameter right here. We still need to find all the other parameters. So for that, again, we simulated various experiments and we arrived at these kind of two main experiments. One of them is called the single cycle assay, and it's a single cycle of replication. So what you do is you put in enough virus in your assay that all cells will simultaneously become infected. And so you put all this virus in, so at least one infectious virus per cell. What that means is all cells then will absorb one or more infectious virion, and that will cause those cells to then start virus production, basically almost at the same time. And so the cells, you can see the start of virus. So if you count the virus as a function of time, it will correspond to the average behavior of a single cell. So you can look at the single cell level what the replication kinetic looks like. And so you could see here at first the virus is absorbed, and then you could see the start of virus production in the cells that produce virus first. And by the time the last cell, the cell that's slowest to start producing virus, you're over here, then all cells are producing virus, and the virus is being produced and it's being matched by the rate at which it loses infectivity. And then over time, then the cells begin to die, and you can see that as a decrease in the virus, and then you can see the decrease in the virus concentration as a function of time, and that's your C parameter that you could recover again if you kept measuring. In the multiple cycle assay, you infect with very, very few virions. So I shown just a little single one here, and that will infect just a few cells, which will go on to produce virus, which will go on to infect other cells, and so on and so forth. So you have multiple cycles of replication. And so while the upper plot, the single cycle, allows us to see the kinetic at the single cell level, so the replication kinetic at the one cell level, the multiple cycle allows us to see the kinetic of spread. So the timing between generation of replication, and also basically the speed at which you're able to get into new cells. And so the replication at the multicellular level is in the multiple cycle assay. I've put a little purple star here to indicate what would roughly be for influenza, what would roughly be 24 hours. And so 24, 48. So this is around here. So you can see kind of the first cycle ends around here. And you could see here that same moment. So you had the first burst, and then you're starting the second burst of cells as the secondary cells become infected. So that star corresponds to approximately the same time in the upper plot. So it's just a time scale marker, that little star. Okay, so that's our experiment. So let's see how we marry the experiments and the math together. So you have the model at the top here still. Here, this is how we introduced us. We were able to estimate C. Now the eclipse phase, so this is the single cycle. So when all the cells are infected at the same time, and the black dots here, everywhere the dots are actually real experimental measurement. So here you could see, again, that shape I was describing in a previous slide, and approximately here would be the mean length of the eclipse phase. So some cells will start production earlier, some cells will start production later, but on average, cells will be starting virus production post-infection roughly at this time. So in this case, it's around 6 hours post-infection. So that's our eclipse phase identified. So we now have this parameter and this parameter. We can get the other parameters based on multiple rounds of replication. Now, mind you, if we had kept measuring even longer on the single cycle, we could have seen the dots start dropping off, and then we could have gotten this infectious lifespan. It's rarely done on a single cycle assay because people have to take so many data points at early time points that they get very tired by then. And so by the time they can come back to do the rest of the experiment, like the next morning, we're well into the decay. So we miss the exact timing of the decay. So that's why it's not usually done here. But on the multiple cycle assay is usually when we get to actually see when the cell begin to die. So the width of that plateau is approximately the infectious lifespan. Here again, we recover the clearance rate. Now, this flattening, which is here and here, it's when the virus production is matched by the clearance. So because we have the clearance already, we can determine the virus production from the height of the plateau. And then finally, the upslope, it depends on all the parameters, but because each of the other parameters depended on another feature of the experiments, we're now able to recover beta. So this is how basically we can map each parameter to a specific feature in the graph. Now, we don't actually do it in this kind of one equation, one unknown type of approach. We do just a Markov chain Monte Carlo to more generally estimate the posterior likelihood distribution of these parameters based on the shape of these data points, based on all three experiments. So we estimate based on the combined data of all three experiments being simulated at the same time to do this. So here's an example of an application to this. So this is mapping the genotype, meaning a specific mutation, to phenotypes, meaning what parameters are affected or what replication cycle aspects are affected by that mutation. And this is based on in vitro data in flu. And this is work between my group at Ryerson and my collaborator, Guy Boivin at Université Laval and his team there. So if you look here at the parameter estimation in black, and I call them black virus and red virus, but if you want to know what they are, the black virus was the pandemic 2009 strain, and in wild type, and then the red one is the H275Y mutant. It's the one that confers resistance to Tamiflu or oseltamivir. So here what you see on the left is the single cycle and the multiple cycle experiment. Here the single cycle is only in infectious virus measurement units, and here the multiple cycle is both in infectious virus measurement unit, but also in QRT-PCR in the empty. So the empty symbols are QRT-PCR, so total virus count, and then the solid symbols are the PFU. And from that, we're able to estimate all these parameters. So you can see here, for example, the time to virus production is actually longer in the mutant than the wild type virus, but the production rate also higher in the mutant than and in the wild type than in the mutant virus. But overall, their reproductive number is roughly equivalent. And you could see that fitness is recovered because the mutant is more infectious per virion than the wild type virus. So at any rate, you could look at the specific for that strain, but we're able to extract all these individual parameters. Now, we also were able to verify that we can extract them properly. So here's a prediction of a competition experiment. So this is not actually a fit, this is a prediction where they've put in a 20% wild type, 80% mutant, or 50-50 or 80-20, where they're like poured in and then they are made to duke it out. And this curves, the dashed line predict how much virus there will be of each of the two strains. So this is a prediction based on the parameters we extracted, and you can see that we do a good job at replicating it. It's a bit far here, but don't forget that there's error bars here on the 80-20. It's hard to get exactly 80-20. But you can see it's actually, so it is within error basically, but I'm not showing the error bars, so I apologize for that. So you can use things like that to estimate not just the impact of a specific mutation. You could compare two strains, but you can also put antiviral drugs. So you can imagine having the wild type and the wild type plus drug, and then you could actually compare the two. So let's go ahead, for example, one thing we were able to do is by running this with two different strains. So we had the H275Y mutation, but also the I223V mutation. We were able to compare, firstly, how these two strains would do against the wild type in the absence of therapy. This is at the top, or in the presence of therapy, and this is at the bottom. And so this is wild type is black here and mutant is red. So you could see that they're roughly matched in the absence of therapy, but in the presence of therapy, of course, the drug-resistant mutant will dominate. Same story here. And then you could see in particular, if we compete two mutants, we could tell which one would dominate both in the presence and absence of antiviral. So we could see that the H275Y mutation was a much better fitness than the I223V. So if we wanted to know kind of what strain could win in a competition, we can do that from the parameters. So this is basically a very powerful method that we could apply to other viruses as well. I wanted to spend the 15 seconds I have left to talk to you about another project that we've completed now about getting better data to quantify infectious virus. Virus samples are quantified because, as you know, only a fraction of virions in a sample are infectious, and they're usually the amount of infectivity in a sample is usually quantified by the number of infection it causes through what's called a plaque assay or a TCID50 assay. We think this assay has a number of issues. In particular, it can be hard to count what's a foci and what isn't, especially there's like edge effects. We think the TCID50 is a bit more robust because it's one or zeros, but it's a bit of a weird measure because you say you have one TCID50 of infection. What you would want to know is how many infections will the sample cause per milliliter. Now, in the interest of time, I'll skip how we do this. This is the team who has worked together to come up with this. This is the paper, and I just want to show you two things. Number one, when you take our measurement, so you still do a TCID50 assay, but you estimate the final quantity you estimate is no longer TCID50 per mil, but specific infection per mil, that's SIN per mil. And you could see that it's very consistent with the PFU, which is a much more biologically useful measure. So you could do a TCID50 assay, but get a PFU equivalent measure, which is more biologically directly usable. You know how many infections to expect per milliliter of your sample. And if you don't know how to measure it, you can just go to our website called MidSyn. You enter your assay. You can even do that on your phone from the lab. Click submit samples, and you'll get what you get as an output is the posterior likelihood distribution of the virus concentration. So for example, here it's 6 to 6.16. So it means that you have 10 to the 6 specific infections per milliliter of your sample. So in the interest of time, I'm going to wrap this up right here. If you have any questions, I will be sticking around until 4:30. So I'm happy to answer additional questions about specifics about your virus and how we could apply that to your virus or about the measurement technique. Thank you very much.
Thank you. My guess is that you'll have people wanting to walk through those slides in detail in the question session. I certainly want to see them in detail during the question session. So I appreciate that very much.
Happy to do so.
Can I ask a quick question?
Okay, well, we're getting set up for our next, yes, go ahead.
Thanks a lot, Dr. Beauchemin. It's great seeing you talk. Just, I may have missed it at the beginning. This, the cell culture in which you're testing this is just some epithelial cells, right? It's not a combination of immune cells and that type of thing. It's just target cells.
It very much depends on the virus. When we've had influenza, it's usually been just MDCK cells, sometimes transfected with alpha-2/6 receptors. We've also had Calu-9, but basically, yeah, usually it's the cells either do form a tissue or do not. We've done with HIV, these were free-floating cells. In this case, they were immune cells. But we've never had the host cells plus an immune response, but it would require a modification of the model, but it would be possible to do so, of course.
So the clearance is essentially a saturation of the target cells.
No,
right? The fall off of that period.
The clearance is the loss of infectivity. So the virus over time loses, the surface proteins lose stability and they start to pop out so that the surface receptors start to degrade. And so the virus is no longer able to enter the cells as easily.
Okay, thank you.
You're welcome.
So our second talk is by Austin Baird, Modeling Whole Body Response to Infection and Associated Acute Inflammation, Clinical Treatments and Outcomes.
I appreciate you all letting me give a talk here. This community is really, really great, and I'm really honored to be able to give this talk. I'm a new hire here in the Division of Healthcare Simulation Sciences. So a lot of what I'm going to present here is kind of related to healthcare simulation, or when I say simulation, I don't mean computer simulation, I mean a training environment to train clinicians on how to properly care for a patient. And so a lot of these models are kind of based around the larger physiological outputs of the patient instead of maybe fine-tuning, like that excellent talk we just saw, fine-tuning some of the interaction models at the cellular level. So let me kind of go over just how I model physiology. And I want to start by just talking about BioGears a little bit, because that's the platform that I use to model inflammation and infection. And it's been in development for a long time. It's had support through multiple DoD contracts. And it's, you know, continuing here at the university to be developed. And it's a really nice platform for healthcare simulation because it has a really robust API and software architecture on the back end that kind of lets you dynamically interact with the patient. So because all of that work has been done already, in the background, it's a really nice place to execute some of these models, because then it allows you a piece of software that's able to plug into a mannequin to train a clinician on how to identify some of these subtle cues in physiology. So this is just some of the functionality that's here. It's going to be very similar to like maybe the presentation, the really good presentation on QMOD in the last week's talk. But I want to kind of get into what is the engine. And we kind of think of it as a middle out approach to modeling. We kind of start at that tissue cellular level and then build out towards a body response. And computationally, we want to make sure that BioGears had low computational overhead, that it runs very rapidly. So these aren't sort of high fidelity Navier-Stokes approaches. But they are something that's able to be run faster than real time in order to support training. And so what does this look like modeling? And I'll try to leave out a lot of equations, but basically we have a physics-based model of the cardiopulmonary system that's a lump parameter representation of that circulation. And so what does that mean? It means that we represent circulation similar to how you would solve an electrical system. So we have things like capacitance to represent vessel flux and shape change. We have resistances that kind of impact how flow is handled. And then we're able to calculate things like pressure, volume, and flow rate in this system. So we kind of we have this trade-off, right, between increasing fidelity and computational cost. And so that is kind of what informs how you want to create a physics-based calculation of the cardiopulmonary system. So what does this look like in BioGears? We can kind of stand up this circuit. And then when we decide that we want more sort of spatial resolution or more complexity for a given organ, it's written in an object-oriented way, so we have all the computational ability to kind of expand how the fluid is handled in a certain organ or region. And so you can kind of see this with the renal system. Was updated to kind of have a much more complex physiology representing sort of what we call a lumped nephron system that has glomerular filtration, reabsorption, and then all of this is, you know, giving us pressure, volumes, flow rates, and bladder, and the tubules, and things like that. And then on top of that physics-based fluid circulation, we then stand up compartments, and this is going to be sort of able to inform what we think of as like a traditional physiologically based modeling framework. And so these compartments are set up hierarchically. They include a lot of the data that's underlying in the circulatory model, and you can kind of just see how we break it out. Obviously, the kidneys right there on the representation on the right aren't the more complicated kidneys, but the totals that are listed on the left are consistent. So we have things like you can ask for lung volume, and then it will kind of drill down into each of its child compartments in order to request what is the volume for each of these compartments. But then you can also request data of things like maybe I want the pulmonary veins volume or flow rate. Maybe I want something like the glomerular filtration rate or the reabsorption pathway, flow rate. So you can access all of that data through these compartment structures. So it's a really nice framework to start modeling some physiology. And so substances have standards and definitions. So it allows for sort of code-agnostic way of transferring substances maybe from one model to another or from one engine to another. So these are built in a generic way. They're not specified specifically for BioGears. You could take our XML files and then port them over to a different model. So this is an example of what acetoacetate looks like. So we template these in XML formatting so that you can kind of have a human readable ability to edit these things. So you can see we have things that tag clearance, and then clearance that's tagged then is able to be used in a model underlying in the C++ code. So that's kind of the nice benefit to creating these sort of top level hierarchies. And you saw that in QMod. A lot of places are moving towards this, CellML, SVML markup languages. So being able to sort of template a model in XML and then have that be kind of shown underneath is something that's really nice in portability and extensibility. So let's talk about inflammation and trauma that leads to inflammation. So in BioGears, we wanted sort of develop an acute inflammatory response model. And we wanted to do this sort of as a response to infection, so leading to sepsis. And so now this is a lot better because now you can see the circles that are being referenced. So basically we have our AIR model with the green circles, and then the light blue circles are sort of feedback models in BioGears that currently exist, things like glomerular TGF mechanisms, nervous system models, things like that. So that's the other nice thing of implementing something in BioGears is you kind of get this framework behind it, and you don't have to reinvent the wheel with a lot of these things. So you can kind of see the solid arrows are indicate upregulation of some of these effects, and the dashed lines inhibit them. So the inflammatory inflammation and tissue damage will lead to symptoms of the response. Usually this is modeled by just a general Emax. As damage worsens, resistance on these tissue pathways, so you can see in the purple box, number 2, those resistance pathways are going to deteriorate and you're going to see a fluid shift. And this is going to lead to hypovolemia, hypoalbuminuria. So like the proteins in the blood that generate that osmotic pressure gradient are going to start shifting as well. So nitric oxide in this model is going to start also exerting an additional hypotensive effect by breaking down that endothelial barrier. Hypertension is going to be induced. Hypertension is going to lead to baroreceptor feedbacks, resistances, heart rate changes, and glomerular feedback is going to lead to urine output reduction changes. So the baroreceptors eventually are going to become overwhelmed, and you're going to see the SVR kind of plateau as a result of some of these things. So inflammation doesn't just happen from nowhere. So we also have a pathogen invasion that can induce this inflammatory cascade. And this can also be induced from other things, some things like thermal trauma, hemorrhage, severe hemorrhage and tissue damage. All of these things have been shown to create inflammatory responses in the body. There's also some role of inflammatory responses in the body in homeostasis, but we don't really get into that in this model. But we're going to create a model of the infection. The pathogen's going to originate in a certain tissue. It's going to interact with local immune mediators. And then eventually it's going to break down that local tissue barrier and start spreading systemically. As it enters the circulation, then our AIR model is going to start responding to that infection. To kind of bypass that pathogen model, a burn trauma is going to just directly lead to that inflammatory interaction with the patient. Just a little bit of details of how does that tissue flux happen in BioGears, or how did we implement it? And this is pretty standard. So we're just going to have this change in flux be due to a change in pressure and resistance, right? But in this model, our pressure is going to be a result of hydrostatic pressure, but then also osmotic pressure. And that osmotic pressure is going to be a function of total protein concentration in the blood. And then there's going to be some reflection coefficients and changes to the albumin concentration that are going to sort of induce a nonlinear response to travel into the tissue compartment. So you can see on the figure on the right is an example of a tissue circuit that's connected to every major organ in biogears. So this isn't just going to happen in one location, it's actually going to be localized regionally based on how much is this resistance impacted for that specific organ. And then we have a lymphatic return pump to sort of return the albumin back into the systemic resistance or systemic circulation. So what do we see with sepsis? So first thing we wanted to do with our sepsis model is sort of quantify it clinically so that we can see things like severe infection, induce clinically relevant markers for a sepsis diagnosis. And some of these things that we broke down, and this was in collaboration with some other ICU clinicians here at UW, but we wanted to make sure that sepsis was classified by its traditional sepsis 3 definitions. This includes things like a reduced blood pressure, an elevated respiration rate, an renal organ failure assessment score, which is going to be quantified by urine output, and then things like a really low MAP, a really high serum lactate that can lead to septic shock classification. So we did this to make sure that when this model is used in the context of healthcare simulation, it's going to reflect maybe what a patient would be responding to this infection. And so you can see on the right there, this is all data taken over 48 hours of infection in biogears, and you can see a lot of these responses that are defined by sepsis 3 behaving in a way you might expect clinically. Now there's a lot of work to be done validating this model, either at the cellular level, at the tissue level, at the nervous system level, and then also clinically at the clinical level. And right, this is why these things take so much time and are so much work. And it's hard to just say generically, oh, this model is the best and it's always going to work. I think I always want to be really, really upfront about validation efforts and how important they are. They're just as important as constructing the model and getting the outputs that you may want. But let's talk a little bit about the AR progression. So basically, you can configure an infection in biogears a few different ways. You can configure it as mild, moderate, and severe, and then you can also configure it as a specific tissue bacterial amount. So we kind of give you the option computationally. So this helps with developers who are creating a simulation using this model. They kind of have an easy means to manipulate the patient. And so you can see the different responses between these different bacterial amounts. You can see the patient is not responding well for the severe. You can see the green curves. Blood bacteria levels are increasing. Interleukin levels are staying high. Tissue integrity is staying low. And when tissue integrity stays low, from the previous slide, that means that there's going to be a corresponding fluid flux into the tissues due to that tissue integrity breakdown. So now maybe we want to parameterize our patient population, because a lot of times, and what you'll see when at least when we were talking to clinicians, is that you have these very patient-specific responses to sepsis. So you might give the same person with what they might think of as a similar infection the same treatment outlines, but they might not survive, whereas another patient will survive. And so we want to think of this as maybe some stochastic variability within the parameters underlying the cellular model. And so I know I haven't described what K6M is and X66, but these are just parameters within the AIR model that's going to lead to a more robust response or a compromised response by the patient to the bacterial levels that are in their body. And so when we sort of parameterize these things this way, sort of susceptible, which we're calling green, which means that the rate of phagocytosis by tissue macrophages, that's the KPM parameter in our model. And our model is detailed in a publication we put out a couple of years ago, so you can see all of the mathematical equations there. So, and then a hyperinflammatory model. So interleukin-6 is going to remain really elevated, especially during the initial trauma. And then immunosuppressed, the purple. So these graphs on the right are all for the same level of bacterial infection from the initial onset. But you can see wildly different patient responses due to that same infection. And so we kind of want to start thinking about, well, you know, especially in simulation, if you want a very realistic sepsis simulation or training application, you need to make sure that your patients are going to respond in very unique ways. And so maybe this is one way we can get about that. You can see the blood volume in the hyperinflammatory patient. It really, really decreases. It crashes much more heavily, although it has the same endpoint after 2 days as the susceptible population. So you can see the interleukin-6 is really expressing itself readily there in that model. And so now once we have a model, maybe we want to construct something by seeing how it does. And this is the nice thing about developing it in BioGears. We want to see how the patient responds to clinical treatment. So things like administering antibiotics, administering fluids, administering norepinephrine in order to control and modulate the mean arterial pressure. And so we found this study when we were collaborating with our ICU clinicians, and they're like, hey, why don't you try to just run the model? And we're not trying to say anything about BioGears being used as a clinical decision support tool, but what we want to see is that it can quantify and classify a patient response in a fairly realistic way. And obviously, as we validate this model more, maybe we can move towards that. But this was just let's see how the urine output responds and how the mean arterial pressure responds when we run it through this clinical trial that has been done previously. And so these are some of the results from that trial. And so first off, we just wanted to make sure, and I didn't talk about our drug model, but the nice thing about using BioGears is that there's a drug model there. And so we could implement an antibiotic that could possibly buffer this infection. So you can see on the top left graph, that is a PK curve for piperacillin tazobactam, I think. And then you can also see the area under the curve for saturating the patient and the antibiotic activity and the blood bacteria level. So you can see that this antibiotic is going to respond really well and decrease the blood bacteria to help the patient recover. And then on the right is in green, where we had early goal-directed therapy, but we didn't include vasopressin. And then on the right, we included vasopressin, but had reduced fluid. So basically, we were targeting the patient here based on their mean arterial pressure. And so norepinephrine was going to be used in order to sort of modulate that pressure. So you can kind of see the cumulative fluid is really a big takeaway here. So there were, 1.5 liters about more fluid in order to sort of have a similar end goal on the map. So you can see the map resolved itself on each of these to be a little bit below 80. So it was able to be sort of stabilized over the course of this. And then you can see the urine output is decreased compared to the early goal-directed therapy. And you especially have spikes when you're really heavily loading the patient with fluids. And so that's the thing with BioGears. There are models that are associated with each of these things. So MAP is regulated by nervous system. It's regulated by all of these things, whereas when we created this and stood up this model of inflammation, the only thing we were targeting was this fluid damage, this endothelial layer. We wanted to see hypovolemia happen in the patient due to sepsis, because that's a very strong clinical marker in the decreased MAP. And then everything else we left alone. And so you're able to get this sort of framework to ask questions like this when you implement a model in BioGears. And so that was kind of some of our motivation. Just really quick, burns, what do they look like? So this is a 10%, 25%, and 40%. So this is all still just our inflammatory model. And you can see the differences in blood volumes and arterial pressures and heart rates. So this is all just leading to similar patient trauma as sepsis, but now induced by something like burn. And you can kind of see the cumulative fluids as a result of that and the blood volume responding. So also, like, BioGears is an open source community. We have documentation. We have a GitHub site if people are interested and want to become involved in the project. It exists. Yeah, you know, we hopefully have a lot of people. You can join our public Slack channel. And you can also just start pulling down the code. It's free. Go download it. If you have any questions, I'm always happy to help. Reach out as to how you can use it and leverage it and where models should be implemented and things like that. So thank you, and the references.
Okay, wonderful. Thank you so much. So the floor is open for questions for either of our speakers.
Hey, Austin, Jim Feckler here. Nice talk, but again, I'm biased as a pro person here for what the work you're working on. Can you send me your slides?
Yeah, absolutely.
I need the references, you know. And then the other thing, and you talked about it. What would it take to use this at the bedside? You know, to feed the data in, and say, you know, if you do fluids, here's the distribution output that are possible, as opposed to if you use a vasopressor at this point, given everything you know. And is that possible?
Yeah, so I think that is a good follow-up to that clinical trial. I put it in air quotes that we tried to run with BioGears. So yeah, there's definite decision points that you can construct because BioGears is sort of a dynamic runtime of the patient. The simulation doesn't just run statically, right? You have a patient who is respirating, and this is kind of why it was constructed this way, in order to put that in a mannequin and be able to sort of have these decision points. But in terms of the bedside, yeah, I mean, you could imagine this eventually, like I said, with the asterisks of maybe after we validate XYZ,
No, I get
each
it.
level of the model. But
It's too soon now.
Huh?
It's too soon now, but
yeah, absolutely. But you could imagine, yeah, BioGears running as maybe you receive somebody in the ICU and then you stand up a BioGears equivalent patient. You configure them with the trauma and the demographics that they would present with. And then you could sort of experiment on the BioGears patient of what, you know, or you could use it as an after action tool or other applications like that. I think that those would be really cool. That is such an awesome application, but I also think you know, we always want to put like an asterisk of there's still a lot of work to get there, and I don't want to overstate how awesome BioGears is. Although I love BioGears.
And I'm a fan too. All right, yeah, cool.
Yeah, but I think possibly, you know, maybe we could get there someday. I think the really important part is how patients present individually, especially with sepsis. And I've encountered this a lot when I talk to ICU clinicians, and you probably know this too, is like sometimes people crash and sometimes they don't. And maybe you're kind of giving them similar treatment to begin with. Obviously, when they're crashing, you're going to treat them differently. But how do we parameterize a patient in order to sort of get that variable response, especially if you don't know their underlying sort of inflammatory response ad hoc, right? They come into the ICU, you're not going to know how does your interleukin behave when you're presented with a bacterial infection. Although, you know, you can see in the model that when we can parameterize something like that, maybe we would have very different patient responses. So I think those are really good questions.
Gary, you had your hand up. Yeah.
Yeah, so great stuff, Austin. I think I'm pretty familiar with your work. You know, so the application for this, I mean, this is the goal, right, is to have this running on the bedside, being updated by data streams and perturbations and that type of stuff that would essentially refine what your parameter space would be to a particular individual patient, because the specification that you have is essentially a wireframe diagram of the hemodynamic fluid physiologic responses to sepsis, in which that they're going to have differential responses based on where your particular inflammatory state happens to be at that particular time, and any sort of genetic or epigenetic predispositions prior to that. So the decision support role for this would be, if it were light enough weight to be running at the box, so that it could be, you know, a box at the bedside, so it's actually being recalibrated or refit with some sort of forecast horizon, you know, as you go to see what's, you know, potentially going to happen. You know, so in the class of interventions that, you know, that we currently have, which are fluids, pressors, et cetera, right, the kind of, you know, complementary approach to that type of decision port is, you know, the DRL AI clinicians that have been coming on. I think a couple of people have made these, which take the, you know, big data streams and, you know, essentially reproduce your circuit with an artificial neural network, right?
Yeah.
And I think that, you know, I think thinking about how this would work in the future is some sort of complementary, you know, hybrid system that utilizes these two technologies together in some way, shape, or form. Because the challenge, as Austin probably knows, you know, with these mechanism-oriented, highly complex mechanism-oriented simulation models for prediction, is that there are too many uncertainties in the real world to actually be as good as a very data intensive environment that can do prediction through correlation, you know. And I think that I think is a huge challenge. We ran into this issue probably about almost 20 years ago with the Immunetrics, you know, sepsis models, where you have a great deal of, you know, of some of the stuff that's in BioGears, but you just can't match in terms of the predictive horizon a data-centric thing, because there's stuff that you don't happen to have in it. So
yeah, and I think that's a good point. And you know, like I said, when you're thinking about parameterizing patients, you know, this is where a lot of that physics-informed deep learning models really come to the forefront, where they're able to sort of predict certain parameter spaces based on the solution set. So they've run them a lot with the Navier-Stokes equations, where you maybe have a pressure flow field, and then it's able to sort of back calculate some of the flow rates or some of the parameters in the underlying model. And so, because I mean, ultimately, you know, machine learning is an optimization problem. So,
right,
you know, you could think of sort of a hybrid thing where the machine learning model is sort of optimizing for certain parameters in the underlying mechanistic model, and then maybe it feeds those parameters into that model, and then you can interact with that model via decision support or something like that.
yeah, I mean,
yeah, there's got to be a combination of the two, and it seems like there's a lot of really cool work that could be done in that space, I think, that hasn't.
Well, for the machine learning, you need to have the data, which is why they can come up with an AI clinician that manages fluids and pressures, because there's a ton of data on that. Yeah. Now those things can't explore anything that you haven't already tried. So you cannot put into that particular system or even do anything about that particular system in terms of new drugs, combinatorials, anything that you already haven't done, right? So the mechanistic component is to explore new things. But the challenge is, if you're trying to optimize existing therapies of which you have a lot of information about, then that's what the machine learning is for. And just one comment about like the Navier-Stokes equation parameters and all that type of stuff. Those are dynamic too. The patient
Yeah.
does not have a consistent one during the entire hospitalization. It can change on a, minute by minute, hourly by hour standpoint, and that
Yeah.
those are not, linear progressions. The fall offs are essentially what account for patients who crash, or differential tipping points that lead to thresholds of responsiveness and blah, 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 all this type of stuff, so,
Yeah.
but very interesting work.
Yeah, and a lot of those models, they'll find localized node, and they're not able to predict some of those large crashes and things like that. That's like what Tesla's having issues with, right? With self-driving cars. They just can't.
Well, that failure mode has to be present enough in the data for the AI to find it, right? And I think
Yeah.
when you're dealing with data sets like Mimic, et cetera, where you have, which are as large as you can possibly get, there is enough consistency about that. So for the vast majority of things, it will work. And again, this is part of the problem with respect to how AI is presented, because I didn't catch whether or not you're a clinician or not, but the clinicians
I'm not.
among
I'm
us.
a PhD in applied mathematics.
Okay. So the clinicians in us know that the actual number of patients in which there's a great deal of uncertainty about how they're going to do is actually relatively small, right? So if you search across the entire space of all the patients and how they're actually going to do, most of the results are going to be trivial, where there's not any sort of uncertainty about what's going to happen. The probably less than 10 to 15% of patients have some degree of uncertainty with respect to how you think they're going to respond. You either know or you don't know. That's
Yeah.
Well, great,
Yeah,
great stuff. Very interesting.
Yeah, it's a great discussion.
Okay, other questions more for Catherine or Austin? I definitely have questions for Catherine. I was going to ask for Austin, and maybe it's already been said, but I wanted to clarify it. You showed the example of your predictions as for your different patient models. So you had your immunosuppressed patient, your hyperinflammatory patient, and I guess one of the questions which would come back to Gary's, and maybe you already answered, was which of the metrics that are easily clinically accessible help you stratify the patient fastest? Which ones tell you that this is an immunosuppressed patient as the first? Which ones diverge most quickly of those? Because the interleukin-6 is a hard thing to measure.
Yeah.
But blood pressure is easy to measure. So things that are easily accessible clinically, which are the ones that give you the stratification of the patient the fastest? And maybe Gary will say you already know that beforehand, which is fine. But I just, from the mathematics, which ones tell you the fastest?
I mean, that's a nice question for an inverse problem, right? Because when you measure blood pressure, right, you're getting the response after many layers of maybe models or the physiological response building to that. And so there's a lot of cool mathematics around inverse problems. I think there's still, I don't think it's a solved field, although I'm not super familiar with it. In order, how do you take blood pressure and then tell you something about interleukin-6? Because there's multiple parameters that are going to result in a blood pressure issue, right? So it's not just interleukin, it's nitrous oxide, it's also the baroreceptors, or how the nervous system's interacting with that patient. And so there's, we could take the blood pressure and then we could parameterize multiple things in order to sort of induce that response in the patient. So,
Right.
So then how do you quantify a probability of each one of those things? What's their significance?
Right.
What's their most likely response in the patient? I've talked to somebody that said that they're working on sort of in vitro work to start measuring some of the septic markers and inflammatory markers in a patient, but I think it's still a day or two to get that data. So you draw the sample and then you get it a day later. And so that's never going to work as a real-time parameterization rate of the patient.
Well, you can get turnaround in a couple of hours.
Oh, can you?
Okay. Certain things, and people are working on these bedside mediator profile things.
Yeah, absolutely.
Testing type of thing. I think to go to the inverse problem, it's not possible because the non-uniqueness of the mapping is so broad in terms of identifying, the various mediator combinations that can lead to a particular phenotype. It's not possible. I think moving forward, and this is something that should probably be integrated in any sort of continuous control process, is you want to take advantage of the perturbations that you put into the system, right? That determine what your differential responsiveness of the system is, and that will lock down your parameters a considerable amount. We do this clinically by giving things like fluid challenges or testing, turning the vent up or down and that type of thing to see where they happen to be, and there are a whole set of, things that you can potentially do, provocative testing, that would determine where this goes. So like, for instance, in your particular system, giving different fluid challenges at various points in their resuscitation, and then determining where in their forecast horizon they end up, right? That would help you refine where the, what the responsiveness may or may not be. So this is the loop between the mechanistic component, right, which you couldn't do this with a data-centric thing, where you would develop some sort of perturbation evaluation component to refine what the differential responsiveness happened to be at that particular time.
Yeah, and I think that brings up another good point with the sort of data-centric models, is the reinforcement learning AI, and how do you train that? Because when you have like the mimic database, it's not dynamic, it can't interact with it. And so actually one of the things I think would be really cool is to create a reinforcement learning AI and point it at BioGears and just have it start messing around with the patient in different ways and learn how to do certain things, right?
That's actually our work, is that what we're doing is we're using model-based DRL for cytokine mediator management, right? What we don't have is the physiologic component. Now, the virtual AI is a DRL program, and what they've done is they've inferred what the counterfactuals would be based on data matched patients who didn't get a particular therapy, right? And because they have enough to do that, as people who know about this would realize that there's a potential flaw in that, because there are other factors that that counterfactual is not. It requires several levels of inference and is a statistical probability. But yeah, so I think, like, for instance, running DRL on synthetic data on BioGears would be very interesting compared to what a with DRL mimic-based AI does.
Yeah, that would be a really cool experiment.
Yeah, but our particular interest is specifically in using DRL on cytokine mediator level models, high resolution models of sepsis, to come up with multimodal mediator based therapies. So, so the synthetic data generation issue is a very interesting one in the biosphere, which here I think, briefly alluded to that as well. So.
Yeah.
Okay. Maybe Catherine, can I ask you to come back to some of the slides that you had? Because as somebody who tries to build models of in vitro viral replication, I'm always stymied by the way viral quantities are measured. The TCID50, as you point out, is a problematic measurement. Others are not so good. And so any insight you could give us into better ways of quantifying virus in a useful way from the point of view of modeling would be very much appreciated.
I have been muted and unmuted. Can you hear me?
Yep.
Oh, sweet. Okay. So, I mean, my screen still says that I'm muted, so everything's frozen. I will speak, and I don't think I can share my screen because the previous speaker is still sharing their screen. But to answer your question.
Sorry, Catherine, you should be able to share now.
Okay. Like
If you can't, If you can't, it may be a Zoom crash, in which case I would ask you to exit the Zoom and restart it. Because
So I can try to restart, but it did say that it's because the previous speaker was still sharing.
right, but I fixed that, so you should be able to share now.
I'll go ahead and leave and come back then, and hopefully that works.
First try sharing, because you should be able to.
Okay.
But if you can't, then we can try.
Okay, I'll try again. Yeah, it's still warning me that I cannot.
It doesn't look like I'm sharing anymore. It looks like I still can share. I don't know if I need to like give away presentation. Oh, there we go.
Can you try now, Catherine?
Yep, I will. Again, and hopefully this time it works.
One thinks that one has encountered every Zoom problem already after 2 years of this, but there's always a new one.
Oh my God, it still thinks I can't. I am not
So
sure
then it's a problem with your Zoom session. So
okay, so let me quit and I'll come back in.
And while we're waiting for that, if there's another question for Austin, please. I didn't mean to cut off a valuable discussion at all.
Oh no, you're good. You're totally fine. I didn't want to like hog the question and answer session, so
there are no rules. It's an open discussion.
Okay.
Hi, Austin. I have a question. Do you guys hear me?
Go ahead.
Yeah, go ahead.
Perfect. I think I asked this at some point, but I'm just like refreshing the question now. Like, is BioGears like, let's say all the features of it, let's say, are still like, let's say, self-contained within BioGears, or there are like, let's say, plans to make it interact with other systems, like through, let's say, co-simulation or through some interfaces that would allow it to let's say, work with other simulators or with some hardware in the loop. Like, are there some current examples of that, or there are like plans in the future to make it, let's say, work, let's say, in tandem or in synchronization with other systems to achieve, let's say, a higher, more complex features, let's say?
Yeah, in terms of hardware in the loop, BioGears has been used, like I said, for healthcare simulation. So it's been used in robotic mannequins. So it can sort of interact with hardware in various ways. In terms of interfacing with other systems or models, yeah, I mean, I would love to have that happen. So like I said, there's sort of this generic substance data, right? But you can imagine maybe making other things more generic, things like standing up the cardiovascular system, or things like, you know, you could pull out pieces, but the problem is, and then you could serialize those in and then stand up the C++ code. The problem is that everybody needs to agree on like what is the, I mean, you could think of like a standard, which is the finest grain resolution of data. And then you could have things sort of select certain bits, but you need a data transfer between the two that is consistent between the two. Or otherwise you're just like feeding garbage into something. If that makes sense. I'm not trying to be harsh.
Yeah.
Or saying any other system is garbage. But yeah, I would love, I mean, I think that that's where we need to work towards is how do we define that data? How do we define a system that is portable and interactable? Like even an example with like CellML or cell, you know, how do we serialize CellML implementation into BioGears and then let it specify an organ that's being damaged or a larger system that maybe isn't templated in that model? You know, so how can it interface with BioGears? And I think these are all really cool questions to ask, and I think they need to be asked because when these things live in isolation, they just die. So, and that's also why the open source community is really important as well, because you want people to adopt BioGears and you want people to adopt all the things that are open source and use them, right? This is how things like Node.js become ubiquitous in JavaScript programming, right? Because Node.js is open source and Google released it or Facebook or whoever did, and now everything is written in Node.js and there's a set, clear, defined JSON format file that people can serialize in. So that's a much simpler example. They're not trying to simulate the human body, but you know, I think there's a lot of value in open source development, and I think there's a lot of value in asking those questions. So hopefully we can keep asking them.
Yep,
Okay, let's come back to Catherine's presentation, and then we'll have time, I hope, for maybe another question or two.
Sure.
Cheryl, if you can hold on for just a sec. Thank you very much.
So I think your question was about how infectious virus is measured. Yeah.
I had that, and there was a second question too, which was, but let's start with that, because I wanted to understand the quantification, because I think that every model that somebody does of in-host viral infection and spread and immune response starts from the viral load.
Yeah.
And if we don't understand how it's quantified, what that quantification means, you can't go anywhere.
Yeah.
So I think you're addressing one of the most critical problems in this kind of modeling and experiment.
Yeah, well, it became clear at some point our model got accurate enough combined with the right experiment that we started pushing against actually the limitation of the experimental measurement and the uncertainty they introduced. And that's why we started, our attention got drawn to this aspect of things. So if we're talking about virus, you can measure kind of total virus. Usually people do with qRT-PCR, and it's basically counting a specific protein. So you've got to have your probe, and it's got to be matched to whatever part is unique enough. And there are various problems with that, but the important problem is infectivity. And because ultimately you don't care about virions that will not cause infection, you care about the ones that will. And even that is a bit of a complicated discussion, because if a virus cannot cause infection by itself, but it could cause interference in infection, then they might still be relevant. But let's abandon that for a second and talk about specifically infectious virions, those that are capable of causing infection kind of by themselves, in and of themselves. So they have fully capable virion. And people cannot count those. We can't count them by visual appearance. We can't count them by mass, or we have to. The only thing we can really count is the infections caused. And the issue with that is the infection caused in a person might not be the same number of infectious cause in the cell culture that you've chosen. And so even there's a big problem where there's a disconnect between what we count and what there would have been as a number of infected cell of infections in the patient, right? So it's not an ideal way of counting. There's even situations where the infection takes place, for example, in Calu-3 cells, but then you actually are counting the infectious virus in MDCK cells. So you don't know how many infectious virus you had at the time of the infection, when the infection was in situ, right? And so that's a bit of an issue. But if we leave even all of that aside, ultimately what you're counting is not infectious virus, but infections that took place. So if, for example, you have an infectious virion, and the infectious virion is fully capable of an infection, but stochastically fails to cause an infection, you will not count it, right? And so there are limitations to that. But so let's go ahead. So if you have an infectious virus, what you can do is you can count the number of infections your sample will cause on average when deposited into certain cells. So that's how many caveats are actually there in the counting. And so one of the assays, the plaque or focus forming assay, where you actually deposit your sample at some dilution, increasing dilution, and then you let it incubate for maybe an hour. You have to rinse it off. You put an agar on top. And so the cells that are originally infected by the inoculum they deposited take on the virus, start producing virus. But because of the agar, the virus can only squeeze to the immediate neighbor. And then when they produce virus, it can only squeeze to the immediate neighbor. So by counting the number of plaques and that you observe, either through antibody staining, this is what I'm showing here on the left, but you can also do that through like cell death if the virus is particularly lytic, and then you look for holes. So you do a health stain dye, so like crystal violet or something like that you see here in the violet. And you'd count the holes, and that would be how many infectious virions you had in your original sample, assuming that each plaque is actually one infection caused to begin with. But it does get complicated. If you look in the zoomed in version, you see this very large patch here in the left, but you also see this tiny patch, and you see all these specks. And the question is, are the specks just error? Are there small plaques that fail to fully develop? and so on and so forth. So by how much might you be undercounting your virus? You can also see all this brown edge here. And so these are all virions you've not counted probably because of the meniscus and because of the cell culture's edge. So there are issues with that. From that perspective, I find a TCID50 assay by concept to be a bit more easy to follow, because in a TCID50, you deposit the virus in the cell culture, and basically you have like 1 million cells in these wells, and you wait long enough that the infection is either fully, so there's no agar overlay. You deposit the virus at various dilutions. So this is each row here has a different dilution. This is undiluted at the bottom, and then 0.2, so that's a 1/5 dilution at each one. And you basically go and see what fraction of wells get completely consumed. So it's not like counting individual infection. You either have had an infection or not. And the assumption here is that if you had any infectious virus, then it's usually enough, the production is usually enough that it will eventually overtake your entire well. So you're really counting, did it happen or did it not? So it's a 1 or a 0. But from the fraction of wells that get infected at each dilution, you can infer what's called a TCID50. So the place where that curve crosses the 50% point, that's the dilution at which it happens. So your virus concentration in TCID50 units is 1 over that dilution. But having 2 TCID50 per mil is not a useful measure. Like biologically, what I want to know is if I put 3 milliliter of that stuff, how many infections will it cause? That's what a biologist really wants to know if they want to plan out their MOI. So this TCID50 is kind of not very useful. And the other thing is it's actually not quite accurate. So 1 TCID50 does not correspond to 1 plaque or 1 infection cause. And so it's not biologically useful. So we went back and basically using Bayesian inference, we transform what would be, for example, this is a random simulation here where you could see how many virions are received. And if you pay attention to this one, it's a 1 here. And so you'll get a full infection. So the well will become white as opposed to purple for the uninfected well. And so by counting kind of what fraction of wells are infected, you can infer what must have been the concentration of infection causing unit in the original sample. And so that's what we're doing. So instead of using this old school TCID50 method that was required when the method was introduced in the 1930s, is when that method was introduced. Well, since then we've had some better computers. And so we can really estimate the concentration of infection causing units in the original sample. And we call that measure SIN for specific infection. So from your good old TCID50 assay, you change nothing about what you do in the lab, but instead of using Reed and Munch or Spearman-Carber, you go and plug in your numbers on our website. And I'm going to show it again right here. You go on that website, you enter your well volume, your starting dilution, your dilution factor, the number of dilutions you have in total, the number of repeats you have per dilution, and then you say how many wells were positive. And then you just click submit sample outcome, and you'll get one of those graphs that I just showed you. So a graph like this that gives you the estimate of the concentration, but also the uncertainty on that estimate. So you also have error bar on the confidence you have in that estimate. So that's a tool that you can use right now. There's the website. I can give you the URL right here. So this is the URL. So you can go and use it in the lab from your cell phone or from the computer, whatever's available in the lab, and just you can bookmark that.
So may I ask a question a little parallel to what I asked Austin, which is the dilution, the serial dilutions take time and they cost some effort, and one could imagine that the doing tenfold dilutions is pretty coarse measurement. When you do your analysis, do you find that if you did, how much accuracy would you gain by doing 5-fold dilutions or 2-fold dilutions rather than 10-fold dilutions in the TCID basic protocol? Is it worth doing that?
Well, I would ask you to read our paper. We have done it. So we have done a measurement. And what we find in particular is that you are better off with more dilution, so more fine dilution, than you are with more repeats per dilution, which is unfortunate because one is a bit more work than the other. The difference in accuracy can go, I think, from plus or minus 0.3, I think, at dilution that people typically use to about 0.1 in TCI. Like, so if you look here, so we've got 0.1 here, so that's at a 10-fold dilution. I think you could increase that. People typically decrease that to 0.3, and that actually is that's a third of a log, right? So it's a lot of space used up on the error bars here if you do something like that. And I don't think it's advisable because you've already put so much effort in the experiment itself that it's really silly to cheap out on the very last step of getting your accurate measurement. So to me, I say, sure, the additional dilutions are more work, but the experiment is worthless if you can't get the level of accuracy that allows you to tell the difference between two strains. Because the more accurate your viral titer measurement is, the more accurately you can tell apart two strain properties. So was it worth doing, right? If you want your research to not be underpowered, then that extra accuracy could be the difference between a non-significant result and a significant result. Can you afford to risk it? And so that's where I would say the cost really is. If you've already done the experiment, a few more dilution is, I would argue, always worth it. The other thing is, I've not really published on that yet, but we have evidence to suggest that a lot of the stochasticity in inter-experimental variability, or even like within experiment replicates, the uncertainty is dominated by the measurement and not by variation between cell cultures. So if you have 3 wells and you could see the errors between the 3 dots, the 3 repeated measurement at that time step, most of that error comes from the measurement uncertainty, not in fact from variability between the 3 wells, which we thought was pretty remarkable. This will eventually be published, but I'm just kind of scooping myself here. So I think it's definitely a big bottleneck right now in accuracy.
So I don't want to monopolize, but I had one more question for you, which was at the very beginning, your whole cascade of measurements starts by having to measure the decay time of the virus.
Yeah.
And you had the interesting result that essentially the decay times of the virions and the decay times of infectious virus were quite different.
Yeah.
Which was already quite profound. But I did have a question about how you did that particular experiment, because you said you did it in the absence of cells. And is it done in supernatant from the cell culture so that whatever RNases and things are present, or is it done? What is the culture condition in which you're measuring the viral decay?
Yeah, so to quantify the virus, we need the cell, but to observe the decay, we don't have cells. So it's only the supernatant. So we'd have the virus sample in the same medium as our incubation medium or our maintenance medium, which are often the same. And so you just have the virus sample in its normal medium. And what you do is at routine interval, you'll draw, for example, let's say you have 3 ml of it, you'll draw maybe 0.5 ml at every sample time point. And then you'll reserve that sample and break it into 2. Some of it you'll process through PCR, and so those are at these points, and some of it you'll process through a TCID50 or a PFU assay to count how many infections the sample can cause per mil. And because the surface protein of the virus get degraded over time, as you repeat these measurements with further time intervals, so basically you've let that sample sit at 37 degrees Celsius for, let's say, 24 hours or 48 hours or 72 hours, you go back to the same sample and keep drawing new samples out of it to measure. And you will count fewer infections caused per milliliter of that sample.
Right, but I guess my question was, what is the medium in which the virus is sitting?
Well,
because the cells themselves could be secreting something which degrades the virus actively.
But it does. So that's a good point. So the cells, okay, you're right. So the cells could be degrading the virus. So as part of the infection, in this case, there are no cells, right? And so there's no cofactor of infection that might be degrading the sample. The sample itself is whatever you stored it with. And so if when you took it out of the cell culture, you buffered with something, or it's up to the manipulator, right? The person who chose to do the experiment. We try to reproduce the normal experimental conditions without cells.
But you start out with the virus growing on top of the cells. Mm-hmm. You take the fluid that is on top of the cells with the virus in it, you reserve it, and then you watch the virus degrade. Is that the experiment? So in fact, the medium is the supernatant from the primary experiment.
Correct.
Okay, so whatever virus degrading factors are present are going to be there also in the are going to be in the superdata, although they may not be refreshed by the cells underneath them.
That's right. So they're not being refreshed. And you can imagine that those factors would be different early in infection than they might be at the end of the infection, right? And so that would always be true. So it is possible that if there are such factors and they're secreted in large quantity over the course of the infection, this could become a time varying quantity. And people have never looked at that, mind you.
Right. So that was what I was trying to say, that maybe this that you do. Okay, thank you. That was really helpful. Okay, we're slightly over time. Does anybody have one final question for our two speakers?
So I had a question for Austin Baird, and I'll try to make it a quick question. I had a somewhat lengthier question, but what is the range of timescales that BioGears works along, like the minimum and the maximum of the orders of magnitude of time variance, and does it deal with circadian rhythms?
Great question. Yeah, so basically we have a standardized time step within BioGears that we use to solve sort of our systems of ODEs and the circuit responses and cardiopulmonary systems. So that is on the order of like 0.05 seconds, so very short, so that we can simulate the dynamics of contraction and relaxation of the heart. And then in terms of how long a patient can be sustained and are there circadian rhythms, that's a great question. Actually, we did, so a BioGear simulation can run, like I showed, 48 hours. It is for rather acute patients like that, 48, 72 hours. You could possibly run it 4 or 5 days. The issue is that we actually you would have to like feed the patient and give them water, and you'd have to also have them sleep. We have a sleep amount.
Yeah, that's just.
But it actually but right now it doesn't actually impact the hormones in BioGears. So it's more just it impacts how the patient digests glucose and how the patient responds to what we call a psychomotor vigilance task. This was to support a specific project like a year or two ago. And so we didn't consider things like some of the hormone fluxes, because honestly, some of those models we actually don't even have in BioGears yet. So then it would be a matter of kind of, but we keep track of sleep. We keep track of how much the patient has slept and how much the patient has been awake. And then right now, all we do is impact glucose metabolism as a function of that. So it'd be very interesting to think about how those things impact some of the sort of the long wave dynamics of some of the hormones and things like that. But that's definitely a model that would be really awesome to integrate. But in terms of weeks and months, BioGears is not probably a great platform because we have to solve the system at that small of a time step. It makes that, so right now it runs at about maybe 8 times real time. So for every 1 second of real time computationally, it'll solve 8 seconds. And so then you can kind of extrapolate how long, 80 hours can be simulated, within 10 hours or something like that. So the longer and longer you go, the more and more computational requirements there are. It is threaded in a way that you can stand up multiple patients and they'll all run concurrently, but it's not multi-threaded for a singular patient instance. So there's probably room for speed to increase in BioGears. So that's also something to keep in mind.
Thank you.
Okay.
Very interesting.
Okay, well, I thank both of our speakers for staying and having a great conversation. And we have a lot of time zones in the room today, so I appreciate everybody joining from England all the way to Japan. It's quite a scope of time zone. Thank you all so much, and I look forward to seeing you next week. And please do send me your suggestions for speakers or volunteer yourself. And with that, I'll call the meeting for today to a close. Thank you again. Goodbye.
Thanks.